so fast. But anyway, just a little background. Of course, we're talking about AHC and its related conditions, which are, of course, genetic diseases of the gene ATP1A3. And for those of you that have kept track of this field, you know that ATP1A3 is a gene that codes for a protein called the sodium potassium ATPase, and we abbreviate sodium Na and potassium as K, so that's why it's NAK ATPase. But this is basically an ion pump, as illustrated in this cartoon in the middle of the screen. And what it is pumping in all cells, it is removing sodium ions from inside the cell and replacing them partially with potassium ions. And I say partially because there's 3 sodiums are removed, but only 2 potassiums are replaced. And because each sodium and potassium ion has a single positive charge, there's a charge imbalance. And that leaves a net negative charge inside of cells. And that's a really important physiological principle that underlies the phenomenon known as the resting membrane potential, which is all cells kind of carry this slightly negative voltage across their cell membranes, and that helps the cells maintain certain properties. Nerve cells, or what I'll call neurons, have a particular form of this protein that's encoded specifically by ATP1A3. And this is important for setting up ionic gradients of sodium and potassium that come into play when the neuron needs to fire its electrical signal called an action potential. And I'll show some examples of action potentials that we recorded from AHC neurons in a moment. So just moving forward now, we think, based on a lot of indirect evidence, that the ATP1A3 mutations responsible for AHC and probably related disorders are probably largely mutations that knock out the activity of this ion pump. So we call them loss of function mutations. And what that would do, we predicted, was it would alter the ability of the pump to create this resting membrane potential. It might affect the ability of the nerve or neuron to make action potentials. And if we were really careful, we might be able to measure directly the activity of the ions, the ion pumping activity directly. Because of this difference in sodium potassium transport, we should be able to see a net positive movement of ions in the cells, and that should create a small electrical charge, something called pump current. So that's the sort of the basic science lesson for the day. What we've been doing for the last few years in collaboration with Kevin S. at Vanderbilt is taking either skin cells or blood cells from kids and young adults with AHC and telling those cells that they need to kind of be converted back to a more embryonic state where they were capable of growing into different types of body cells, a state we call pluripotency. And this was a breakthrough in medicine that earned the Nobel Prize for a Japanese scientist, Dr. Yamanaka, a few years ago. And his group determined that you could reprogram body cells, like skin cells and blood cells, to go back into this pluripotent state. And then once they're in that state, we can then direct them to become other cells, such as brain cells or heart cells or muscle cells. And then we would be able to study those rather than trying to biopsy those organs from a patient with a disease we're interested in. And so Kevin S.'s lab has succeeded in generating quite a collection now of ATP1A3 mutation carrying iPS cell lines. That's the name of these pluripotent cells. We've been able to generate several mutations that are common in AHC, including the two most common mutations, D801N, which is right here, E815K, which is here, G947R, which is, I'm not finding it, but it's in here somewhere. Right, it's not showing up. Oh, it's right there. And we have a couple of others that are interesting that we haven't done much with yet, but we're working on those. And the goal here was to collect cells from more than one patient with each mutation, and that's taken a while, but we've succeeded now in generating quite a library of these cells. And now we're in the process of turning those cells into neurons. Just so you can see what these cells look like when they're in the pluripotent state, here they're microscope images of the cells that have been stained with different markers that inform us that they are in a pluripotent state and are ready to be directed into different growth patterns, into nerves, into heart cells, whatever we choose. So we've been making them into nerve cells, and we really didn't get this going until my lab moved to Northwestern, and we were able to work with a very talented stem cell biologist in the neurology department, and we worked out quite a few details that allow us now to generate brain cells, neurons, from these patient-derived pluripotent stem cells. And so these are just some pictures of work that was some cells generated by Christine Simmons in my lab, showing you the progression from early day 5 to day 14, day 21. They start out not looking like very much on the left at day 5, but then they begin to sprout these long strands of tissue that resemble nerve fibers, and that's what we have in this last slide. And here are some other pictures of them. We also demonstrated that the neurons do have ATP1A3, a critical step to make sure that we're not studying something that's completely irrelevant to the child or the patient. And then this is the slide I started with on the big screen. This shows you some really beautiful pictures of what really are neurons with extensive branching patterns. These long tangles are sprouts from nerves that are reaching out to try to connect to one another and form networks of nerve connections, very much like they do in a developing brain. So what can we do with this? Well, we were fortunate into having some very healthy cell lines from two unrelated patients, both that had the G947R mutation. One was a female and one was a male. We had different independent lines of cells that we could then turn into neurons. And so over the course of really about 9 months or so, Christine just slowly slogged through making many batches of neurons from these two patients. And then we were studying them as she made them. Chris Thompson, a very talented scientist in my group, would then study the neurophysiological properties of these neurons. And he really wanted to measure all of the things we think define how a neuron functions. Now, probably most of the people on the call don't know what any of these terms mean. I did talk about resting membrane potential early, and so we want to measure that as an index for how well the pump works. Initially, we didn't think we could measure the pump current directly, but Chris had a major breakthrough in February and was able to do that. And we, in fact, held up our manuscript while he spent 2 months generating these data, which are really, really beautiful, and I'll show them to you. And then we measured quite a few other features, including how well the cells were able to form and generate action potentials, which is really the sort of essential job of a nerve, to generate this repetitive firing activity, this electrical firing that signals to other nerves and to collectively make the brain work. So just to show you sort of what we think is really the most impressive feat so far, is these squiggly lines that you see here is a small electrical current that's being made by these neurons. And we know this is coming from ATP1A3 because of two things. First of all, when we have the cell bathed in a solution which is devoid of potassium ions, there is no current. And that's consistent with the fact that in the absence of potassium, the pump cannot work. Now, normally there's plenty of potassium around, so the pump can generate activity. But in the laboratory, we can start with completely absent potassium and then throw in and bathe the neuron in a very high potassium solution. And when we do that, during the time represented by this bar, suddenly there's current. And that current lasts the entire duration of this exposure. And then we wash the potassium out, go back to zero, and what we see is flattening of this line back to nothing. Then we repeat this experiment in the presence of the well-known inhibitor of ATP1A3 called wabane, and we try it again, and what we see is we've turned off some of the pump by this inhibitor. So we can actually measure the height of these peaks and deduce the amount of current generated by the pump, first in normal healthy cells, and then in ATP1A3 mutant or AHC-derived cells. And here what we see is that the difference between the high potassium in the absence of wabane and in the presence of wabane is lost. So we have lost this extra bit of current that is present, which we think is directly ascribable to, or is the first direct evidence that a mutant ATP1A3 has lost ion transporting activity. The pump is broken. We didn't really have direct evidence for this before, but now we do. Also, this gives us a new measurement that we can use to determine if drugs or chemical compounds that we've identified can boost this blunted current that we see in the patient-derived cells. Can we restore that to normal? So this one slide with these squiggly, chatty lines is something that we were just incredibly impressed with and proud that we were able to do it. By the way, these currents are really small. This line here, labeled as 5 PA is the amperage, the units of measurement here, which are picoamps. Pico is 10 to the minus 12th amperes. So it is a trillionth of an ampere that we're measuring here. And that's pretty small. So here's just sort of a summary of data that is shown in sort of a statistical way that is illustrating the amount of pump current that we measured in normal cells, which are the gray ones, and the AHC-derived cells, which are the red circles. And so you can just sort of get a sense for that the amount of current that we're seeing is much lower. And if we look at specifically the wabane sensitive component, that's also lower. There is a bit of wabane insensitive current. We don't know what that is. It may be another form of sodium or potassium transporter, which is commonly there, but we don't need to worry about that. So anyway, again, we think this is the first direct evidence of impaired sodium potassium ATPase from a mutation in ATP1A3. Now, knowing that, what else could be wrong with the neurons? And again, we look very extensively at resting membrane potential, which is really an essential property of nerves, allowing them to function properly. And again, with knowing that there was impaired sodium potassium ATPase activity, which is really required to charge the membrane electrically, we thought that the resting membrane potential, or RMP in this case, as illustrated on this slide, would be lower in or be more negative, I should say, in control healthy neurons than it would be in the AHC-derived neurons. And in fact, that's what we saw. So in one clonal line, one cell line from a female patient with the G947R mutation, we saw this very large 10 millivolt, that's an electrical term, 10 millivolt difference in the resting membrane potential, making the resting membrane potential in the AHC neurons much less negative. And that's a sign of impaired sodium potassium ATPase. Now, we weren't happy with just showing that from one of the lines. We actually grew up the other line and saw exactly the same thing. And we also made multiple rounds of neurons that's sort of encoded in the shading of the symbols, and we also saw consistent effects there. And then lastly, to cap it off, so we know that this isn't a female-specific issue, we looked at a male patient and his cell lines derived from who have the same mutation. And looking at neurons from this case also showed the same difference, a less negative resting membrane potential. And knowing that now, a less negative resting membrane potential, we assumed there might be a difference in the ability of these nerves to generate the electrical signal that we call the action potential. And so that was the next set of experiments, and without going into great detail, what we did here was we stimulated the nerves with an electrical input of current, and then we watched to see if the membrane voltage changed. And when you see these spikes of membrane voltage, that's what we refer to as an action potential. And sometimes you get one, and sometimes you get more than one. And when we looked at the parameters, variety of parameters are determined in this case. We can either hit them with a single stimulus and sit back and watch what happens over time. We can also turn up the stimulus, turn up the current, and inject a higher amount of current. And what we should see is an increase in the number of action potentials over time. And so when we do that, we see that for the normal or healthy neurons, we get between 1 and 2 action potentials all along the way with different levels of current injection. With AHC neurons, the best we can do is 1, and sometimes we don't even get 1, so it averages out to be less than 1. So here we have an example of there's a diminished ability of the neurons to fire. And that suggests that that goes along with impaired pump function and impaired resting membrane potential, and is again a strong clue to what is the essence of the defect in AHC. And again, this brings us additional measurements, both the resting potential and the ability to fire action potentials. These are additional measurements that we can make before and after applying a drug or a compound that we think may rescue the fundamental defect in the disease. And so this has been, like I said, this project has been going on several years, and I think we finally gotten to a point we thought we'd be at a little earlier, but this has been a really important last few months of bringing it all together. So I'll mention a few other things. That was I know that seeing a lot of data on a Monday night is not what most of the people on the call are used to doing. So I will just list a few things that we're doing now that will just to share with you our excitement about what is possible going forward. So I mentioned in the beginning that the Dr. S's lab has made several additional ATP1A3 mutations and several pluripotent stem cell lines. They're constantly refining their technique and trying to make new mutations. And I know that they're engineering some of these mutations for which patients are not available, which is really another breakthrough in technology. They are also, as we are, trying to develop a technique that we can correct the mutation and convert it back to the normal DNA sequence in the cells. And the reason we're doing that is not because of a therapeutic goal, although that would be a really lofty goal. We're doing it so that we can eliminate the genetic background in the cells as a contributing factor to all of these electrical abnormalities that we're seeing. And that's a challenging thing that we think we need to start doing on a regular basis to ensure that our measurements are really direct measurements of the fundamental defect in the neurons and not a reflection of some difference in genetics, of the difference in genetic background of the people that we're studying. We are poised and are preparing, we've generated neurons for other mutations, 2 of which are listed here, and one other one that I don't list, and that we are preparing to investigate. Now that we've hammered out the basic principles of what we want to measure, the work should go much faster, and we should have a much more rapid turnaround of results now that we've sort of worked out the fundamentals. Now, the neurons that I showed you in all of these prior studies, they come from a particular type of neuron, which we call excitatory neurons. These are neurons that stimulate in a positive way the activity of other neurons. But they're not the only class of neuron in the brain. There is a class of neuron that inhibits the activity of other neurons. And of course we call those inhibitory neurons. And they may also be important to study, and we have actually developed tools to study those, but the procedures are very slow and very laborious. And we're trying to find strategies, as I know Dr. S's lab is as well, trying to find strategies to speed up the generation of inhibitory neurons. Right now, it takes 80 to 90 days to make these neurons, where the excitatory neurons are ready to go in 3.5 weeks. So that's really slowed us down quite a bit. There is a new technology that's been reported from a group of scientists in Singapore, and they will be sharing some of their protocols with other labs, and hopefully some laboratories here, including ours, will be able to get that. The other thing we want to do is we want to look at neurons that are releasing dopamine into the brain. And dopamine neurons are important for movement and are well known for being involved in the disorder Parkinson's disease. There is some evidence from some of the mouse models of AHC that dopamine neurons may be involved. And so one of our other efforts that we're involved in is to use one of those mouse models, which has the mutation D801Y, and this is work being done in my lab by Thomas Holm from Denmark, who came over last April with the mice, and he's actually recently been investigating dopamine neurons in the mouse brain, and we think that's going to end up showing some important differences that might have implications for treatment. So we're also very involved in trying to identify new drugs or new compounds that might boost the activity of the pump encoded by ATP1A3, and we're getting ready now to try testing some of them on AHC neurons to see if they have any effect on the activity that I was talking about before. And then lastly, we want to also use this mouse model to engineer, to test whether drugs and compounds can modulate the behavior of the animals in a positive way, suggesting that those might be applicable treatments in humans with the disease. And then lastly, we are positioning ourselves to be able to look for genetic effects in the mouse because there may be other genes that impact the severity or can lessen the severity of AHC and dystonia and other features that are shown by this particular mouse model. And these other genes themselves might be drug targets. So we think that's a really important area of investigation. So I'm open to taking questions now. I have some other things I wanted to talk about, and maybe as people on the phone call are thinking about questions, I'll also sort of explain why we're having this call today, which is a couple of weeks ago, I talked with Lynn and Sharon about allowing me to write a short article, or help write an article for the newsletter, which came out last month, on the importance of the membership of the AHC Foundation to advocate with your congressional representatives on the importance of making sure that our federal funding for medical research from the National Institutes of Health remains strong. And as maybe some of you know, that there's been some discussions in the Congress and the Senate and the White House, all starting to position and prioritize what should be in the next federal fiscal budget that will take effect in the fall. But there was also concern that the current year's budget was about to run out, and you may have learned that last Friday Congress passed a one-week funding bill to try to give them a little more time to hammer out how the government should be funded now until the end of September. And one of the proposals on the table was to perhaps cut back on federally funded medical research both this year and even more so next year. And I think that would be very damaging, and a lot of people were really worried about that. So I was very motivated and to talk to Sharon and Lynn and to help them put in an article into the newsletter talking about the importance of the National Institutes of Health for funding medical research, and letting you know that rare disease research depends very heavily on the National Institutes of Health. And we as in our lab at Northwestern, in collaboration with Dr. S's lab at Vanderbilt, we have been planning for almost a year now to submit a major NIH grant proposal soon, ideally this summer. And when we heard that there was threats to the NIH budget, we really felt like this was going to be a bad year to try to put that in. Fortunately, the news this morning was very optimistic in that the Congress over the weekend did put together a funding package for keep the government running for the current fiscal year through the end of September. And included in that was the authorization for a bill that was passed into law last December called the 21st Century Cures Act. And that act really was a bipartisan bill. This photo actually shows quite a collection of Republicans and Democrats from many states, maybe some of the states represented on the call, that were really backing this bill, because it really put funds into important areas such as cancer research, brain disease research, and a new area called precision medicine. And I think it's important to see that this bill was passed in the House of Representatives by an overwhelming majority, 392 to 26, and it passed the Senate by an overwhelming majority, 94 to 5, with very few people dissenting because they disagreed with some of the language in there that gave pharmaceutical companies some indirect benefits and that sort of thing. But the bill really had a tremendous amount of funds for NIH to be spread over a couple of years. A lot of that is earmarked for cancer research, Alzheimer's and other brain disease research, and precision medicine initiatives. But there was some other money that is just generally able to support funding from other programs. And so this was a step in the right direction. And this follows basically 10 or 15 years in which the NIH budget was pretty flat and didn't grow at all. And that's really made it really difficult to get new grants funded and move science forward. You know, it's important to realize that and here's some of the additional details from that Cures Act, which shows you how the money is spread across multiple years to fund brain research, precision medicine. PMI is the Precision Medicine Initiative, the so-called Cancer Moonshot program, and some other areas such as regenerative medicine. And so this is really a very important piece of legislation. Now, the fact that Congress put together a spending package over the weekend to include 1 year or partial year of funding, 2017, is a great move forward, but they still have to vote for it later this week. They still have to vote for it to put it into law. And so that's just this year. Then they'll go to work on the next year's spending bill. And again, there's going to be a moment of anxiety among people in medical research as whether or not the White House proposal to trim the NIH budget in favor of other priorities will take hold, or whether Congress and the Senate will maintain their bipartisan interest in continuing strong medical research in the United States, which has really been the world's leader in medical research for many, many decades. And now we're starting to see the rise of investments in medical research from countries in Europe and Asia. In particular, China is on a path to surpass the United States in total investments in medical research in just the next couple of years. And it would be really sad, a sad day, if all of our decades worth of world leadership in medical research were suddenly eclipsed by emerging efforts in China. And I think most of us would feel really that was, we don't want to hand world leadership of science over to China just yet, and maybe never. The other important thing, and here's just some important points that I think are worth dwelling on. The NIH is, although it funds medical research, it turns out that medical research has tremendous benefits to everybody. It has strong economic benefits, and if you do have an opportunity to talk to someone in Congress who represents you, or the Senate, these are really important points. It's not just medical research for a few special interests and drug companies. It really benefits a lot of people. There are over 350,000 people employed today in all 50 states. That's the total number for all 50 states that depend on funding from the NIH for their job. If our grants are cut, we fire people. That's just as simple as that. Another thing people have tracked over the years is what is the return on investment in terms of economic growth, job growth, and other things when the federal government invests in medical research. And there's been some very careful estimates over many years. For example, if you just look at the the Human Genome Project and the area, the burgeoning science of genomics, which, by the way, we would not have the ATP1A3 gene identified in AHC if it wasn't for the Human Genome Project. That was funded by the NIH. And that stimulated not only new scientific advancements, but there are very clear estimates of what this multi-billion-dollar federal investment in science did for the economy. And that has been estimated around $1 trillion during these multiple years. And the reason for that was there's been a lot of spin-off companies, technology, medicines, genetic diagnosis. There's an entire industry of genomics that didn't exist before this investment. And so if anyone thinks the investing in medical research doesn't pay off, they're wrong. There's very good evidence of job creation, new business ventures, and a tremendous amount of economic growth and economic impact in all 50 states. And so those are important points. And I think it's important for everyone to realize that you live in a state, and I don't know who's on the call, and I don't know where you're living, but I know Lynn is on the call, and I know Lynn lives in California. But there are currently 54,000 jobs that depend entirely on this one tiny fraction of the federal budget, which is NIH. Other states are listed here. I'm in Illinois. That's 12,000. There's a document that I could pull up on another screen here that shows all 50 states. So if your favorite state is on here, so I know Dr. S is on the phone, Tennessee, there's a few thousand jobs that are almost 7,000 jobs listed there. But these are data that are widely available, and people have kept track of them. And it's investing in medical research is widely viewed as the most cost-effective thing the government does. It boosts investment in other areas, it creates jobs, and it's really a very important thing. And not only that, the added benefit to advancing health and investigating rare diseases like AHC is just not possible any other way. And so I leave you with a few talking points here. If you ever, and I do strongly encourage you to, tomorrow is the day, they're going to be voting this week on what's called HR Bill 244 to fund the government through the rest of the fiscal year that we're in currently, which ends at the end of September. And then they'll go to work on the next fiscal year, which starts October 1st. And these same discussions are going to happen again. Should the government boost funding for medical research? Should they follow the guidance of the 21st Century Cures Act enacted last December? And all these things are going to be on the table again to discuss. And I think all of you on the call have a vested interest in this. If the NIH remains strong or grows stronger, certainly Dr. S and I will benefit, be able to get additional funds from the NIH to really move the research forward. You know, I think another very important point, which is AHC is a rare disease. Rare disease research just doesn't happen in the private sector. The pharmaceutical industry really can't take the risk with something with a small market value unless there is a compelling reason to. And then usually what those reasons are, someone usually in an NIH-funded laboratory at a university has taken all the risk out. They've done the hard work. They've screened libraries of compounds. They've invented a strategy. They've come up with a new technology. And now it's easier to find investors to take it forward. But it's becoming more common now for academic drug discovery efforts to supplant the efforts of the pharmaceutical industry for rare diseases. And the NIH provides great support for rare disease research. And I can tell you, if the NIH budget is cut, the first thing that gets decreased in priority is rare disease research. It's just the significance is attributed to that is thought to be less impactful than common diseases such as cancer and Alzheimer's and the things we know are facing millions of people. So to maintain strong rare disease research, what your foundation has done over the last few years has really been remarkable, and some of the results I've shown you today in the first part of the webinar would not have been possible without the efforts of the AHC Foundation. Going forward, you know, I think to expand this and to make it much more impactful, we really need the NIH to stay strong, to continue to grow, and for that federal funding of medical research to be still considered to be a high priority and a valued priority by members of Congress and the Senate. And so I urge you, this is a good week to learn how it works to call your senator or congressman's office, and sometimes you'll rarely talk to them. You'll usually talk to a representative of them, and that's okay. They log the call, they make notes, and if 1,000 people called their office on one issue, they take notice. If 100 people call their office, they take notice. And so I strongly encourage you to call, tell your story, have some talking points ready to go about your child's health and your experience with AHC and why you think it's important for the government to invest in research that will help rare diseases such as AHC move forward toward cures. So, well, with that, I will take a break and let anyone ask questions if they'd like. I'll leave these up. So I see there may be some questions that have come in online.
No, I don't see any on the chat window.
Okay, hello, this is Bill. I'm listening. This is Sandy. Okay, so I see, yes. So those are, yeah, those are just, got it. If anyone has audio, they can certainly ask questions, or they can.
Everybody can unmute themselves if they want to talk. And
hi, Ray Baker here.
Hi, Ray.
Hi, I've got a question. Has anybody heard about this cannabinoid oil and any research been done on that? In particular, this stuff called Charlotte's Web.
Well, I can give you some background on that. My lab also studies epilepsy, and it really was one rare form of childhood onset epilepsy called Dravet syndrome that brought the Charlotte's Web to the web and to people's minds. And that has stimulated a lot of interest in medical cannabis for seizures. And there is also a drug company in England called GW Pharmaceuticals that is doing clinical trials on a synthetic form of a component of cannabis oil. The particular component they're doing trials on is called cannabidiol, or CBD for short, and there's evidence that it may have benefits for Dravet syndrome and possibly other types of seizures. It has not gotten FDA approval yet, and it may be a difficult thing because in the United States, of course, there's still a federal law blocking research on Schedule I drugs, which includes cannabis and all its derivatives. CBD is not hallucinogenic, and therefore some people think that it's unnecessary to classify it as a Schedule I drug alongside heroin and other things. But that's what we have right now in our country. Now there are states such as Colorado, Oregon, Washington, I know Illinois has medical marijuana approved, where in fact people can get medical marijuana, and in Colorado, which is where some of these cannabis-derived oils have come from, they're distributed there legally, but they can't be shipped out of state. So now that's epilepsy. I am not aware of any anecdotal evidence, or any anecdotes, I should say, stories of anyone with AHC that has tried any of these products. I'm not endorsing them, and I'm not predicting whether they would have any benefit. And I think at some point, if cannabidiol or other things become approved in a way that allows them to be prescribed, then I think there will be an opportunity to test it. I don't know of any current activities or any planned activities to do a clinical trial in AHC with any of these medicines.
Yeah, because the cannabis oil has been legalized in Wisconsin. However, I can't find any pharmacy that can get it, and my physician, Allison's physician, will not give a prescription. He'll go for it, but as far as giving a prescription, he's kind of hesitant on it.
Yeah. Well, there's a couple of challenges in this, which is there's, again, the way medicines are approved requires good scientific grounding that it will be effective. And, that often requires a very specialized approach called a double-blind clinical trial, where there's groups of patients who are on a placebo and another group that's on the actual medicine, but nobody knows who's who. And data need to be collected in a systematic and objective way, and so that we can really test whether, without bias, whether something works or doesn't work. So, for a rare disease like AHC, that's very challenging because the number of cases available for a clinical trial like that is pretty small in any one place. So it has to be a multi-center, multi-state operation. And given the heterogeneity of state laws right now, it would be difficult to pull that off. But again, I think there's hope that the work that's being done in epilepsy will blaze a trail, making it a lot easier for future rare diseases like AHC to be either treated or tested for their response for cannabis-related medicines. The other challenge is just quantifying and measuring features of AHC that can be reliably and objectively compared between groups. And so that's been a big challenge, and I know every time we get together with meetings, that's a point of discussion, is how best to do this. So,
okay.
Any other questions?
That should do it for me.
Anyone else on the call want to ask a question? Okay, I see Tamara has a question. Tamar? Tamar Edelstein? How do you unmute?
Oh, yes. Can you hear me now?
Yes, I can.
Hi. Okay. So I don't know. First of all, thank you so much. This is very informative.
You're welcome.
And thank you all for the work that you're doing and for bringing all of the, this is very informative. I definitely plan on making some calls this week. so thank you. I don't know if you could actually answer this question now because I kind of feel like a bit of an outsider listening in. My son is actually part of the NIH's Undiagnosed Diseases program.
Okay.
With his primary working diagnosis being AHC. And we are actually currently awaiting his genome, the results of his genome to come back. He just turned 5 years old, and basically we really just have no answers as to where his symptoms are coming from, and it's as close to AHC as possible. If we didn't have as much information as we do, we would all probably just think it's AHC. And maybe it is, and maybe it's just like a variant of AHC that has yet to be discovered. So I guess my question is, once the genome comes back, is there any room for collaboration with the Undiagnosed Diseases program? How can I take this further? What can I do as a parent?
Yeah, so so given your son has a clinical diagnosis of AHC,
yeah.
Do you know, was he genetically tested for the ATP1A3 gene?
Yes, he was.
And nothing came from that?
Exactly.
Okay.
Yeah,
so there has been a search for a second gene. Mm-hmm. And at the last summer's ATP1A3 conference in England, there were some reports that there may be some other genes that could cause a syndrome that mimics AHC. So I think the work you're doing with the Undiagnosed Disease Program at NIH will be very revealing, and certainly we would be very interested in working with them and you to sort out what they find. And they rely heavily on the community of NIH-funded investigators, by the way, to help them sort things out. So if they find something that we can contribute in any way, we would be very happy to work with them.
Oh, that would be fantastic. Is there a way that I can facilitate that? Because, obviously things are complex, and I want to make sure that nothing's...
Sure. Well, no, you feel free to suggest that you must have a physician contact at the Undiagnosed Disease Program.
Yeah.
I would certainly suggest that once they do have the results of their genome, and usually they find all kinds of things, they just don't know what is likely to be the cause. And so I will recommend a couple of things. One, please give them my name, and feel free to give them my email address, which is al.george at geez, I'm so tired. I was going to write it on in the process of putting on the slide.
Okay.
You put it on the slide here. Just need a place to write it. Okay. Let me go back into screen mode. Okay, I kind of scribbled it up at the top of the slide. al.george@northwestern.edu.
Perfect. I just took a screenshot of that.
Okay, perfect. Feel free to give them that information, and you can contact me too if you learn of something and you want some explanation. I think that would be something we'd be able to help you with. And also, I'll also recommend, and again, I know that Dr. S is on the call, and he would also probably volunteer this as well. But again, if it's a new gene that we think may be related to AHC, and there's some strong rationale to do some of the neurophysiology, like I showed earlier in my talk. You know, conceivably, one approach would be to work with one of our labs to create these stem cells from your son's blood, and try to use that as a platform to investigate. Now, that's not something we do in a short term. I mean, these things can take a long time, but it would be an investment in understanding what the gene is doing. So,
yeah. That part was very interesting to us because I don't want to take up more of your time, because of some of the things that we noticed that made his symptoms more severe, specifically mistakenly eating MSG, monosodium glutamate, and that really was horrible for him. So I don't know if it's related at all, but I don't know. I just thought it was really interesting and something that might be worth looking into.
Yeah. Yeah, I haven't heard that feature from anyone else, and it would be interesting to know if anyone else on the call has had that experience of symptom worsening with MSG ingestion.
Yeah.
By the way, I'll just say that Kevin S. At Vanderbilt, my collaborator at Vanderbilt, who is on the call listening but can't unmute, said their lab would be, of course, very interested in helping generate stem cells from your son if it looks like it would help solve the riddle. So.
And who is this that said this? I'm sorry.
Kevin S. Kevin is his first name. S-E-S-S is his last name, and let me see if I can put his email on my thing. I like this interactive mode that we're in.
Yeah.
Jeez.
Something a little bit different about my son, and I guess why the part of the mystery is, for the diagnostic criteria for AHC is that, generally it goes away with sleep, whereas with my son, his symptoms are actually triggered during sleep.
Oh, interesting. So it is very different.
Yeah. But other than that, very similar with some other differences, including severe GI symptoms and whatnot. But anyway. So,
seizures? Any seizures?
We suspect, actually, I'm not sure if I'm saying this right, dichristic seizures. which I understand are very difficult to pick up on EEGs, and possibly also gelastic seizures. But yeah, that's been looked into, and nothing. We don't see anything on the studies.
Okay.
Yeah.
Well, thank you for sharing all of that information.
Well, thank you. I really appreciate this.
Sure. Anybody else? Any other questions?
Dr. George, someone posted a question, can't get unmuted either. Is Artane a drug currently being used to treat dystonia?
It's a good question. I'm not probably the person to answer that. I am not sure. I'm not sure.
Okay.
So I'm watching the message board, and actually Dr. S has responded that yes, it is. Artane is used to treat dystonia. Kevin, I wish you could unmute your line.
Hey, Bill, can you go in and unmute Dr. S?
Let's see. Kevin, try it now.
This is Kevin. Can you hear me?
All right. Awesome.
So yes, Artane is a fairly potent medication that movement disorders neurologists often use for dystonia, and it's fairly easy to get a hold of. It's FDA-approved medication, but it's something that a regular neurologist would not use normally, but it's certainly something that can be used.
Okay. Any other questions? Let's see. What else we got here? So Corey Simpson has a question about flunarizine. She's having trouble being unmuted.
She's not connected to her audio. That's the problem.
Oh.
There's an audio. If you called in on the conference call number, there's an audio button. It's pound something pound on yours. It's everybody's different. I'm trying to unmute her.
Okay. Alternatively, if the question is short, you can type it into the question box, and it can be read. I also see Jacqueline Sanchez is also unable to speak. Again, anyone can type a question into the box and we can see it.
Yeah, I don't know how they're logged on that I can't unmute them. But it says you have to connect their audio. Connect to the audio. No, I just did. Jacqueline just came undone. Jacqueline, you should be able to talk. Not anymore.
Roger, check. If you want to go to bed, use your computer. You're more than welcome to.
Jacqueline, can you hear us?
What's the number to...
Candy, can you hear us? Okay. Well, could it because attendee 13 mic is on? No, you can have 25 people talking at the same time, and there's only 19 of us here.
Yes. Oh, I think I'm unmuted now. This is Corey.
Yeah, I was able to unmute you.
Oh, thank you. Sorry about that. I don't know what I did. My question was, our son is 16 months old, and we started flunarizine probably about 2 months ago. He's only on 3 milligrams at this point. Have you done any research on flunarizine as far as dosing, or is there any possibility of it being done in the U.S.?
I could probably address that to Lionel?
Yeah, go ahead.
Okay, my audio quality is not very good on my end. We have a AHC clinic at Vanderbilt Children's Hospital and have probably 6 children at this point taking flunarizine. The dosing range is all over the place in terms of how much you get per kilogram of body weight per day. Everything from several doses a day to some people take it once or twice a week. So a great deal of research could be done into appropriate dosing, but there really hasn't been done. If the FDA would approve it as a medication, then we'd be able to have it more easily available. But again, like Dr. George's earlier comments, I don't see any companies putting a big push to do that, given that investment. But what we can do, and have done along the way, is we do various very research, cell-based, stem cell-based experiments and neuronal-based experiments. We can use flunarizine as one of the candidate drugs to see whether any of the phenotypes we see could be studied in a dish or in a mouse model. So it's definitely something that's always very high on our list. But having enough patients to do a real clinical trial is probably going to be difficult. And then figuring out what the correct dosing is very difficult. So it's something that we often use. I don't think there's a downside to flunarizine other than potentially your money to buy it from Canada for most of our patients. But there's very little evidence to support its usage in the way that we like as a gold standard. But we probably will never have that level of knowledge just because of all the things we talked about.
Right, and that's what it seems like right now. It's just kind of a guessing game on what might work for him, and just kind of playing around with it. When he goes into another episode, we seem to increase it, but there's no real plan in place, I guess. So. Yeah,
we don't. In the epilepsy world, again, we have so much more information. We have really precise dosing recommendations based on actual data in trial. But we can still, often we're still left with trying as best we can to work on patient symptoms. I think the best of flunarizine is going to help modify symptoms, maybe prevent episodes. I don't think it's going to stop the underlying disease process which AHC represents. And that's the area we think we have to move forward from a symptomatic treatment to changing the fundamental behavior of the cells. And that's why Dr. George's data that he presented today is really exciting. Because it suggests there's a fundamental defect we're seeing for the first time, and we can go after that and actually fix the neuronal function. So, again, I know it's never fast enough for parents. I think they are moving.
No, but it's a great start, and it's exciting for us to hear too. So thank you.
Anyone else with any questions?
Hello? Yes, I just wanted to... Oh, hello?
Yes, go ahead.
This is Michelle Maxfield. Can you hear me?
Yes, I can.
Okay, hello. I heard the caller earlier that said that there was a reaction to MSG, and our son Connor, we avoid MSG. He has atypical AHC and has a mutation ATP1A2. Because it's atypical, but we definitely avoid any MSG. It shuts him down fairly quickly, and that's something that we also do. So I just wanted to bring that to that other caller's attention. That we have learned that.
Yes. So Tamar.
Is it Tamar? Yeah, maybe we can connect or something and just let them know that, because he's atypical, and it sounds like her son or daughter, I didn't catch if it was the son or daughter is atypical, and maybe there's something there. So.
Yeah.
Yes, hi. Yes, actually, it was in a food that I later found out had MSG. So thank you for adding that. And we kind of were detectives trying to figure out what was causing this horrible, several 2-week-long night after night of alternating hemiplegia symptoms for him, and every single night he had an episode, and we figured out that it seemed to be the MSG in a food that he was eating that we didn't realize had MSG in it. So anyway, thank you for adding that. I don't know if I can ask this question now. It was just adding on to the flunarizine question, but I won't ask it now if you have anything else to add.
I don't. Go right ahead. Go ahead.
Okay. So we've been taking, well, my son has been on flunarizine for actually almost exactly a year now. And it's been helpful in that it has reduced the severity of his symptoms, although he still does have symptoms. I think it's worked the way it's supposed to work. My question is does it build up, I don't know what medical term to use here, but does it build up in the system? Should there be a time that we come off of it so that like we can restart it at some point so that it works better when we do restart it? And I don't know if I'm using the right like language here, but like let's say if he does better in the summer, because we noticed that perhaps he may have some type of cyclical nature to his disease and does better in the summer, then should we come off of it or reduce it in the summer and come back stronger in the fall?
Are you speaking about flunarizine?
Yeah. Mm-hmm.
Yeah, I mean, again, it's almost impossible to answer these questions because there's not been rigorous systematic studies about it. There have been some concerns voiced by some AHC experts in both Japan and Europe that sudden stoppage of flunarizine may not be a good idea.
Okay.
And so I would discourage you from doing that. Again, it's not clear, but there's been some reports of sudden worsening of symptoms with a sudden stoppage. And I think that's probably not advisable.
Yeah.
You know, slow tapering maybe, but again, probably if you have a neurologist who is working with you, with your child, it would be really valuable to discuss this and make a plan with you.
I was just wondering if there was any research that says that there is any benefit to coming off of it. I know that with some drugs that's sometimes beneficial, but I don't know if flunarizine, if there's any research that indicates that.
Yeah, not that I'm aware of.
Okay.
So there's a couple of questions here. I'm just going to run through them that are typed in. So Jacqueline Sanchez has a child who has been prescribed Benadryl when she has a full-blown attack, and wanted to know if anyone else has a similar, has been similarly prescribed Benadryl and what is known about that. If anyone has an unmuted line that could answer that. That's not a question for me. I think it's for a question for other parents. While people are thinking about that, Renee Hodes wanted to know what would be classified as a brain disorder. Well, I mean, again, that's a very broad term, and certainly AHC would classify as a brain disorder, anything that affects the central nervous system, including epilepsy and AHC and many, many things. So it's a very broad disease classification. Sandra Raina has asked Dr. S to comment on, if he would, the range of dosing for flunarizine he uses with his patients. So.
Hello?
Yes, hello?
Hi, this is Jackie Sanchez.
Oh, good.
I got through. Yeah, my question is, Danielle, she's 23 years old, and now that she's gotten older, she's having the episode of alternating hemiplegia, full-blown. They come on strong now, like no warning signs. And the neurologist gave her, told me to give her the Benadryl shot, which it does work for about 3 hours, 2.5 to 3 hours. And then she'll get it, she'll come out of the attack. Then she'll just get a little stiff after the 3 hours. So they decided to give her the Benadryl pills, one in the morning and one at night, which it seems to be helping. I wanted to know if you know, if that's something that they've been using with alternating hemiplegia.
Yeah, that would be a good one for Dr. S to mention or to answer if he can.
Mm-hmm.
I just wanted to comment on the Benadryl. My daughter's 18, tough one, and we used the Benadryl over the years for dystonia, and it seemed to help just a regular dose of Benadryl. But then we, our neurologist had completed a reaction with AHC, and I don't remember the reason.
Not sure who that is. We can't really hear you. Are
Oh,
you speaking into the microphone?
I don't think so. Can you hear me now?
Not too well, no.
Okay.
Somebody else? Okay. We have a couple questions on my screen if you look at the monitor.
Oh, okay.
Karen asked her question. See the one highlighted? Can you read that or no?
You know, I just see your, all I see is your paper.
My what?
I see your screen, but I just see the wallpaper, the background. Okay, here we go. Huh. Yeah, I'm not, I don't think I see your whole screen.
It doesn't show up. Okay. Let's see.
Why don't you go ahead and read them. Oh, there we go.
Hold on a second. I'm doing something. See that or no?
I do. I can see that.
Want everybody to see it.
Yeah, so you're highlighting the comment from Karen Venti. Charlotte's Web oil is classified as hemp and is currently shipped out of state. I think you may be able to order it. I think earlier Kevin S. had mentioned in one of his texts that, for example, in Tennessee, I think it's not allowed to. You can't get it in Tennessee. So again, every state.
Hemp is classified. Hemp is classified as a marijuana. It's been defined years ago. For this to work, they have to redefine completely what hemp and what marijuana and CBD are. There's many barriers. For each state's different, but it's illegal in Tennessee to bring CBD in unless it's for epilepsy only, and most states have that provision. So just be careful.
Kevin, did you want to comment on the range of flunarizine dose?
Yeah, so patients, again, go from, it depends, of course, on the age and the weight of the child or young adult we're talking about. And it goes anywhere, I see a high dose would be maybe 20 milligrams a day. Each day, usually twice a day, it's divided. So 10 milligrams twice a day is in the higher end. I've seen people take 5 milligrams every other day. And even some people take 5 milligrams 2 days a week. And it's all over the place, which surprised me when I first started seeing patients with AHC. But it just reflects, again, how little we know about this particular medication. And we don't have even basic dosing ranges. Now, the drug is used for migraine headaches and for blood pressure control in other countries. So there is dosing information for people, but for this particular use, we don't have that information. I generally treat patients with the medication every day because I think it has to be there in a certain amount. I don't understand doing it twice a week, daily medication at a minimum, I would say. So it's,
I think everybody would give you a different answer on this phone.
Exactly. Yep.
So that's a tough one.
Yep. Mm-hmm. I'd love to do a study and show something and then have very standardized dosing ranges that would be helpful for everyone.
Maybe we
is
take one more question.
Artane a drug currently being used to treat dystonia?
Yeah, I think we addressed that one earlier, and the answer was yes, indeed it is.
Oh, the next one. You see the next one?
I see one that says, my daughter has one of the less common mutations. Is there anything to be learned looking at some of the less common mutations if the symptoms are different? And I would answer that, that yes. I do think that there is a lot to learn. We've been starting to study a really unusual mutation. I think there's only 2 or 3 patients with this particular one. Because of the character of the mutation, we thought it would be informative. But certainly if there were unusual symptoms or different symptoms and a less common mutation, it might be valuable to study to correlate what the mutation is, what the symptoms are, and how do neurons from a patient like that behave. So I think, if resources weren't limiting, we would look at as many mutations as humanly possible. So I think we would need to know a little more information about what is not typical, what is different, and what is the specific mutation. So to Karen Venti, who wrote this, feel free to email Kevin S. and myself with a little more detail without divulging too much private health information. Maybe just the big picture would be enough, and we can have a little dialogue offline about that. And the last question here, also from Karen, is the salt seems to be a trigger. Seemed to be a trigger for a while for her daughter, but is not now. That's an interesting observation. And Kevin, I don't have enough clinical experience in this, but is that something you've heard, that salt can be a trigger? I think
I haven't, but there are so many different triggers out there. People are aware of the common ones, but I think in general any stress to your body will bring out episodes. But salt, and we have to need more information. We have such a high salt diet already. What type of salt, how much relative, but I can a stressor.
Okay, well, I think we probably have exhausted everybody. I'm a little tired.
I just have one more question.
Okay.
Hi, this is Stacy Greenwood.
Okay.
We received an email today that Wake Forest wasn't continuing the research, and our son was not needed to come in July because the federal grant had been cut.
Oh.
And I was wondering if you knew anything about that, and just how significant are the federal cuts pertaining to the research that you guys are doing in the groups that you're affiliated with?
Yeah, so I know that Allison Brashear at Wake Forest had an NIH grant that, in fact, she may have the only NIH grant funded for this disease. Is she the physician that your child was seeing?
I think Charlotte Miller was the one who was in contact with our family, but I assume she would be the one who's heading the project.
Mm-hmm. Yeah, I don't know anything about their grant status, so I'm actually going to look in the NIH database to see where they are. But this is, yeah, I mean, they do have an active grant until March of 2020, but it doesn't give details about whether the budget was cut. So they may have had a budget cut, which happens. It's happened a lot. Another reason to call Congress and say, don't do that. Let's keep NIH and medical research strong in this country because there's a lot of need. And again, rare disease research is very vulnerable right now. When NIH funding is cut, rare disease research is first to go. I've seen it happen multiple times in my 25 or more year career, where when the NIH funding levels drop, rare disease just stops getting prioritized. And it's just the nature of prioritizing more common diseases. When there's enough funding to go around, people will appreciate the value of studying rare diseases and how equally important they are and how devastating they are. So I think this is a good lesson. Hopefully, I would like to motivate a few of you, if not all of you, to find the phone number for your congressional representative. There are lots of online resources to find contact information and call them. call their office and say, please fund this bill, and then make sure that NIH is well funded into the future, because we need this. And tell them about your child. Because right now there are very few NIH grants that have been funded on this topic. Kevin S. and I want to have the next one.
So Dr. George, that was a kind of two-part question that I had, is if this bill gets passed, would you then go ahead and apply for the NIH grant?
Yeah, I think our plan is to apply this year. And okay. the current year funding probably won't impact us directly, but it certainly will have impact going forward. Again, the next fiscal year budget is also equally important. Okay. And I'm hoping that, again, we can all call again in late summer when they're starting to wrangle about the next year's fiscal budget. So yeah, I actually sent 3 emails to my congressman today with different messages, and I'm going to call tomorrow morning when I get in.
Okay. All right. Well, I think that we know what we need to do as well is definitely promote this, amongst the families as much as we can, and then a reminder again in August, or yeah,
we'll alert you when it's time.
Perfect. Okay. All right.
Can you just show your screen, Dr. George, if you want to just put up the contact information if everybody wants it, the email again, make sure everybody has it.
Oh, I closed my slides. Sorry.
Okay.
Sorry about that.
All right, we'll send out an email to the attendees.
Yeah, that'd be great. All right. Listen, thank you, everyone. I hope this was informative, and thank you, Bill. That was really great help to have it organized like this.
You're welcome. Have a good night, everybody.
All right. Good night. Take care.
Thank you, everyone.
Good night.
Thank you. Good night.